All right, thanks so much, Lauren. And thank you all in the audience for taking some time out of your day to join us for a little bit of learning about powder diffraction. As Lauren mentioned, I'm a senior application scientist with the Bruker Corporation, and my specialty is powder diffraction techniques. So we're very fortunate that we get to dabble in a lot of different industries. Pharmaceuticals, of course, being one of the major industries that benefits from the use of powder diffraction. So throughout the next 45 minutes or so, we're going to go through a little bit of information about what is powder diffraction, and some targeted examples about what types of studies are currently being done within the pharmaceutical industry. Okay, so we'll start out very briefly with some fundamentals. Powder diffraction is something that I was very fortunate in that I got to do this a lot throughout my education, but it's not necessarily quite as abundant as, say, chromatography or spectroscopy techniques. So a little bit of information about what are you looking for? What does the data look like? And what types of information are you getting out of it? We'll also go into a little bit of information about common sample preparation techniques, which are going to be determined by what types of experiments you're running, as well as how much sample that you have. We'll then end the presentation with a couple of common applications, talking about things such as polymorphism or quantification. Okay, so at its core, we are looking at an X-ray tube here on the left-hand side. Your sample goes here in the middle, and your detector goes on the right-hand side. And what we're going to do is we're going to shoot the X-ray down at your sample, and it's going to bounce off of your sample and go up towards your detector. And what we're going to do is we're going to scan our tube and our detector at very specific angles, and that's going to change the angle of incidence between the X-ray tube, the sample, and the detector. Okay. We've actually put together a little video for this, so let me see if I can find that for you. Giving you sort of a brief overview of the technique itself. Lots of really helpful cartoons and schematics. So for the next minute or two, we'll be looking at that, and then we'll launch back to the slides and dive a little bit deeper. Okay, I think we are back to the slides now. Yeah, I just had to switch over from applications. So that should give you a pretty good overview of what happens during an X-ray diffraction experiment. And of course, like most analytical techniques, we don't generally think too heavily about the math on a day-to-day basis. But I think that for those of you who aren't familiar with the technique, it provides a really, really good overview of where the data comes from that you end up seeing when you analyze your samples. If you're interested in seeing that again, we do have a lot of educational videos that are available on the Bruker YouTube channel. So simply go to youtube.com and search for Bruker Corporation, and you can see a lot of these learning videos. So, all right, so if we come back here to our diffractometer, what we see is, again, an X-ray tube on the left-hand side, sample in the middle, and a detector on the right-hand side. So the system that I really want to focus on today is called the D2 phaser. This is our benchtop X-ray diffractometer. And you will see the same fundamental components that you would see in one of Bruker's larger enclosures, but everything has been made very, very compact. So it's the same X-ray tube, the same family of high-speed detectors that are available, sample rotation also available on all of our benchtop systems, but everything has been really miniaturized and brought in close. So what you're seeing here is about a 2-foot box, so it's about 2 feet in every direction. Completely all in one. So there is a built-in monitor, keyboard, and mouse go in a tray underneath. There are USB ports. There is a network port for connecting this to a building network. Integrated cooling systems. The only thing that you need is standard 110 power. So if you can plug in a computer, you can plug in a D2 phaser. And so what's been really nice about this benchtop system is that we've been able to allow people to do these very, very important diffraction experiments in places that previously weren't possible, either due to infrastructure or space or budgetary constraints. Um, but because we're using the same components as the large systems, you're not really sacrificing anything in terms of data quality or integrity. Okay. Specific to the pharmaceutical market, we do have 21 CFR Part 11 compliance that is available. Not something that every laboratory will need, but for those of you who are in a compliance situation, we do have this available. And then with regards to the quality standpoint, all of the phaser systems are available with one of the Lynxi families of detectors. As I mentioned previously, these are high-speed detectors. So if you haven't done diffraction in 10 or 15 years, the detectors are really where a lot of the innovations have happened. And historically, when I first started collecting diffraction data, we maybe needed about an hour to collect data, um, several hours if you wanted something that was really, really good for publication. With the advent of strip detectors and faster detector technology, now we're looking at 5 to 10 minutes for a quick scan, maybe an hour for a very, very high quality scan that you would publish with. All right, so if we look at some examples of data, this is what we're looking at. So on the bottom, on the x-axis, what we see is 2 theta. So as we talked about in that video, this is going to be the angle that includes the angle of the tube and the angle of the detector. So you have a theta angle on the tube side, a theta angle on the detector side, and this combines to give you 2 theta. Counts are going to be on the y-axis. More counts simply mean more signal, either due to more sample or a stronger diffractor. So what we see here is a collection of peaks, and underneath we do see these little red sticks. So this is now being identified versus a database of known positions for this material. So this is silver bainate. So it's a silver with a long fatty acid to the back side of it. Now, when you're looking at diffraction data, what we care about are peak locations, peak shapes. Are they skinny? Are they fat? Are they symmetric? Are they asymmetric? And then relative intensities between the peaks. And these 3 pieces of the puzzle will combine to give you all of the useful information that you can extract out of a powder diffraction experiment. And I generally like to think about this, you'll hear me say the word fingerprint a lot. And that's frequently what we're trying to do is we're measuring something and we're using the characteristic peak positions and locations and relative intensities to identify one material versus another. Right. So some brief comments about sample preparation. This is our standard sample holder. The size of this cavity is 25 millimeters across by about 1 millimeter depth. And in this video, we'll show you how to prepare it. This would be sort of a normal, what we would refer to as sort of a normal sample, where you have enough material to work with. For pharmaceuticals, this can be anywhere between a quarter gram to a full gram of material, depending on grain size and density. So what you see is that I've got a fairly fluffy powder here. Right. So it really is as simple as you scoop the powder inside of the sample holder, spread it out, flatten it, and run it. Um, Once you get a little bit of practice, you can prep samples, in less than a minute and get them going. So what I'm doing now is I'm sweeping off all the excess just to make sure that I don't have a lot of loose powder sticking to things inside of my instrument. So flat side of a glass slide, we're going to flatten it down, make a nice smooth surface, and then I'm going to smooth off the surface. Now, it's important whenever you're running these samples that you have a fairly smooth, homogeneous, fluffy powder. You don't want to have anything that's kind of crunchy. You don't want it to look like sugar or salt. It should be more like flour or powdered sugar, a very nice, smooth, even powder. No speckles, no sort of um, variations in homogeneous. All right. After that, you simply load it into the system and you're off and running. Right. Another way to analyze samples would be with small sample volumes. So this is frequently if you are developing new APIs, excuse me, if you're developing new APIs or you have just a very limited amount of material because it is prohibitively expensive to make. So typically for something like this, what we would use is called a low background, or you'll often hear these referred to as quote unquote zero background holders. And so this is a silicon wafer, and what I'm going to do is I'm going to smudge just a little bit of Vaseline or petroleum grease on there. So I'm going to actually wipe off this excess. That's a little bit more than I want to have on there. You basically just want to have sort of a bit of a smudge, like if you grab your sunglasses with your fingers. And that smudge is going to be just enough to stick the powder onto there. Now, the sample that I'm working with is actually just powdered colored sugar. Obviously, you do want to make sure that you're wearing gloves if you have, a potent sample or something like that. But that right there is enough. That tiny, tiny little bit of sample. If you have more material, you can sieve it or dust it on top, almost like you're putting powdered sugar onto, a cake or something like that. Flip off the excess by tapping it, and then stick that into your system and run it. So that right there is probably about 3 to 5 milligrams of sample. And that's enough to actually get signal whenever you're collecting a diffraction data set. Now, like all instrumental techniques, more sample is generally better. More sample means more signal. But in these specific situations where you don't have a lot of material available to you, using these silicon wafer mounts can be a very, very powerful way to extract out some useful information, even in the absence of a wealth of sample. Another technique that we can use with these silicon wafers is the analysis of slurries or paste or gels. So if you're doing a formulation that is sort of runny or something like a toothpaste or something that is a suspension, you can simply drop cast this almost like you've got a little bit of paint. So here I've got a silicon powder suspended in a little bit of alcohol. And you just sort of drop that on top of the wafer and you let it dry down into a nice smooth layer. You don't want to think about this as a solvent. You don't want to dissolve the solid matter. You just kind of want to make a suspension. And so if you had a paste, you could use a spatula and spread that around, almost like you're putting butter on toast. And then once you evaporate that off, you can stick that on and run it. You could also run this dry, or you could run this wet as well if you happen to have a higher amount of moisture. Okay. The last sample preparation technique that we see is for things that are potent or hygroscopic or maybe sensitive to moisture or air. So we do have these air sensitive holders available. They are comprised of a base plate and a small capping ring. And you will then protect the sample from exposure or protect yourself from the sample if it happens to be a potent sample by using a thin piece of X-ray transparent foil, almost like cling wrap that you would use in the kitchen. As the sample gets smoothed in, same little trick here with the microscope slide, or if you have a spatula, you can do that. So just flatten the sample in there. We do also have the ability to put that silicon wafer in there. So if you don't have quite enough material, you can do a combination of that. So this is a piece of generally either mylar or Kapton film. I like these sort of pre-punched ones because they're easier to grab. And then that black ring will hold the film in place. Okay. So you've got this nice protective barrier that keeps your sample from getting exposed to too much water or oxygen, or prevents you from touching the sample if it is something that is a potent situation or unknown hazard. And then you would just stick the sample holder in your system and run it. Okay, so a little bit of information about what you do with X-ray diffraction, where the data comes from, and how to prep a sample. Now we're going to navigate over to some of the more applied uses of X-ray diffraction, specifically for powder diffraction and within the pharmaceutical industry. So we'll start with positive material identification or qualitative analysis. So I've used the word fingerprint quite a bit here. And so these are all over-the-counter pain relievers. Probably familiar to many of you who are in the audience. And what we're looking at is qualitatively a different set of peak positions and peak intensities. So what I did is I simply just rated the medicine cabinet at work, ground all these up to a nice little powder, stuck them in the instrument, and collected some data sets. And what we can see is that each one of these has a different fingerprint. So you can distinguish between different um, over-the-counter pain relievers simply by just overlaying the data and comparing it. So qualitatively, we can say that these four materials are distinct from one another. Okay. If we look at one of those over-the-counter formulations, here we're looking at not just an API, but we're also looking at an excipient phase. So these are ibuprofen tablets that are covered in a little bit of a crisp shell here. And when we grind the entire tablet up, what we find is that by matching this to a database of known materials, we see a positive identification for ibuprofen, which are the red peaks here, and sucrose or table sugar here in blue. Okay. And we don't see much else, so no other peaks that are jumping out at us. This explains the diffraction pattern pretty well. So the total, so if we look at the black pattern here, so the whole black trace, that is going to be a combination of both ibuprofen and sucrose. Now, with a little bit of clever sample preparation, what we can do is we could either dissolve off the outside edge, or in this case, we took a razor blade and we shaved off the outside part of the shell, and then we ran just the inside of the tablet. And we see that that's the blue pattern here. And the blue pattern seems to match very, very nicely to ibuprofen. So there's a peak there. This sugar peak is gone. These sugar peaks here are gone. Ibuprofen, all the sugar peaks are gone. You see there's actually a little bit of an ibuprofen peak hiding out underneath that sugar. So once you physically remove it, or you could chemically remove it or wash it away, you can get a nice clean pattern of just the inside. So by doing very intelligent experiment design and a couple rounds of X-ray diffraction, what we know here is that ibuprofen is all on the inside and the sucrose is all in the shell. Okay. Here on the right-hand side, we see this nice little silicon wafer mount again. In the vein of talking about excipients, so this is a capture from the FDA. So different colorants. So here we're looking at the first arrow points to titanium dioxide, the second one points to zinc oxide. So these are frequently used in things like, say, makeups or sunscreens. So if you're looking at inorganic sunscreens, you frequently see that these have either zinc oxide or titanium dioxide. A lot of whiteners in, say, pill formulations or paint pigments or paper paper pigments rely very heavily on titania or titanium dioxide. So if you have a very nice white pill, this may be made more white by predominantly titanium dioxide. So if we look at this, so we have two bottles here. These are from the National Institute of Standards and Technology, or NIST, or rather the NBS prior to becoming NIST. Rather older bottles of sample. Two very, very white pigments. And if we look on the left-hand side where the data is, we see that the top scan in red matches to a database pattern for zinc oxide. And so this time we're looking at the peak locations as well as the relative peak intensities. So those peaks right there around 30 degrees, you can see that the biggest one is around 36, the next biggest one is around 32, and the third biggest one is around 34 degrees. So we see that they're all present and they are there in the right ratio. So pretty good sample here. The bottom pattern is going to be for titanium dioxide. And if you look closely, you actually see that there are two different forms of titania that match. Okay. So this brings us up to polymorphism. So within titania, there are two different structures. Even though chemically they are the same and they have the same ratio, one titanium to two oxygens, you have different structures. Here we see two different forms of acetaminophen, or if you're outside of the US, this may be referred to as paracetamol, particularly in Europe. Brand name wise, you might know this as simply Tylenol, but there are two different packing structures for this. So even though we are looking at the same molecule, how these molecules arrange in three-dimensional space is different. And that can have some very, very strong implications in terms of materials properties. For two of the forms of acetaminophen, one of these, if you look at the bottom one, these are laid flat. So on the bottom right-hand side, you can see that this is kind of flat. So this is an orthorhombic form. This would be very, very helpful if you're looking at compressing these into a tablet, because the arrangement would be conducive to that. Unfortunately, the more stable polymorph is actually the top right one, which is monoclinic. So you see sort of a wave pattern on the top right for those molecules. And this is one of the reasons why frequently when you're doing tableting or formulation of acetaminophen, this would be approached via a wet granulation technique rather than just compressive, because the waveform prevents it from compressing very, very nicely, or you might need some sort of binder to hold everything together. If you are familiar with ritonavir from several years ago, this was an HIV treatment drug where there was a new polymorph that was discovered. And unfortunately, this second polymorph that was discovered was very poor in terms of bioactivity and availability. And unfortunately, it was a lower energetic polymorph. So once you started seeing this in your stockpile, it would rapidly convert all of your batch into this less active form. So there's a lot of work that went into determining how to prevent this. There were studies about, do we do a liquid phase? Is it temperature for storage? Does it need to be stored under very, very cold temperatures? Is it humidity induced? So very, very interesting stuff all around polymorphism, that even though you had the right molecule, the long range three-dimensional stacking played a very strong role in whether this material was helpful or just completely not doing anything. So, another example that we frequently see is called the degree of crystallinity. You may also hear this referred to as percent amorphous or percent crystallinity. Here, what we see data-wise is a sample that is predominantly microcrystalline cellulose, or MCC, so very commonly used as a bulking agent in tablets. And then again, that acetaminophen. So we see, I believe nominally this was about a 5% mixture here. So what's remarkable about this is whenever you look at something like cellulose that doesn't have a lot of structure, it isn't quite, we refer to this as not as crystalline or amorphous or poorly crystalline. So this is represented by the shaded in area in the teal color and the light green color. Not a lot of very strong diffraction. right? So previously, up until this point, if we go back one slide, you see very, very nice sharp peaks. Right? That is a material that has long-range order that gives rise to that diffraction phenomenon. So here we would see that the acetaminophen does have those sharp peaks, but for the microcrystalline cellulose, you just end up with this characteristic broad, blobby, non-flat background. So that part of the sample would be referred to as an amorphous or a poorly crystalline material. So what we can do is we can actually look at the ratio between um, your amorphous phase and your crystalline phases and give a quantification of um, how much of my signal is crystalline or sharp diffraction peaks versus how much of my signal is broad or from an amorphous phase. So another example here would be, so how low can you go? Empirically speaking, we've gone down to less than a quarter of a weight percent. So in quantification, things are always done in weight percents when using X-ray powder diffraction or XRPD. So here we have an overlay of a sample that is all MCC or cellulose and a sample that has a 0.2 weight percent blended acetaminophen. So we see a very, very, very tiny amount of signal here, a very small amount of signal at 26 degrees, so the pop out insets. So you can see that it's possible to make out these small loadings, say you were looking at doing a hot melt extrude or a freeze dried or spray dried or a lyophilized sample, and you're trying to prevent your sample from becoming crystalline. We can detect small amounts of crystallinity down to less than a quarter of a weight percent. But it helps if you know where the strong peaks would be likely to show up. Right. The final thing that I want to talk about is cluster analysis. So as we mentioned before, with the advent of faster detectors, with things like the D2 phaser, with these faster detectors, we're finding that more and more, instead of just getting one piece of data to work with, the speed of data collection is allowing people to collect much more data than they used to, which can sometimes result in a little bit of a data glut. So one of the big pushes in recent years is sort of this machine learning or large data set handling, um, especially as computers have gotten faster and more powerful. So within the Diffract EVA software suite, which comes with the D2 phaser, with all of the diffractometers available from Bruker, is the ability to filter these large sets of diffraction data into similarities. So we sort these out based on families. So on the right-hand side, you can see a bunch of different scans. So these represent, I think, 18 to 20 different samples. And you can see that some of them have some similarities. There's some areas where you see peaks that are similar to each other. Um, but if I didn't want to sit down and go through each one of these one at a time, one by one, what I could do is allow the cluster analysis tools within EVA to sort of filter these out into broad categories and then decide which one of these categories do I need to look at first. So it's a good way to process these things. So if we look at the left-hand side here, this is a dendrogram view. This is basically a family tree. Or if you remember back to biology class back in freshman year of college, you do cladograms, and you would sort things out based on which species is related to another. So much like a family tree, the closer these things are connected to the bottom, the more alike they are. So siblings would be much closer than, say, first cousins or second cousins or third cousins. Um, Again, keeping with the left-hand side, we see a box drawn around the yellow cluster. And if we look at the bottom and we compare all three of these members of the yellow cluster, we see that there are very strong similarities between this. So peaks are about in the same locations. We have about the same relative intensities. There are some differences in terms of background, in terms of peak shape. The black pattern has a little bit sharper peaks than the red and the blue one, particularly if you look at that 24-degree region. But because this is comparing all of the data points across all of the data sets, we have a really, really good confidence that these are either the same material or at the very least very, very closely related in terms of data. On the right-hand side, we actually see these broken down into a three-dimensional cluster. There are XYZ coordinates that can be moved. You can zoom in, you can zoom out, you can turn that clustering uh, around, sort of help out with visibility. And now we're going to look at the blue cluster. So this blue cluster has three members that are very close to each other and one seeming outlier. So even though they are very similar to each other, within this family, there is one person who's just dances to their own drum, right? Marches to the beat of their own drum. And so if we highlight that one down below, what we see is that three of the samples, again, much like the yellow cluster, are very similar to each other. Similar peak positions, similar peak locations. But if we look at that peak right there around 20, okay, we see that it is just a little bit different. There is one extra peak around 20 degrees that is sticking out. And that's what's causing that one pattern to sort of stay outside of its own little cluster. So maybe this is a recrystallized starting product, or maybe you've precipitated out some salt, or maybe there's that particular batch ended up with um, something dropped inside of the vial. So it can be very handy if you're looking at just saying, are all of these batches the same? And if not, great, that's the pattern and that's the batch that I need to go back and look a little bit more closely at. Okay, so that's all we have to share with you today. We are going to switch to questions. Hopefully you learned a little bit of something. But I think we're going to turn this back over to the moderators and see if we have any audience questions. So if you haven't had a chance to ask anything, on the right-hand side of your screen, you will have a Q&A panel. So feel free to ask us anything about powder diffraction, and we'll get those questions answered.
Fantastic. Thank you so much, Nathan. This has been awesome. And I do have a couple questions for you. So why don't we dive right in?
Sure.
For the first question we have is for QC confirmation of polymorph, could you compare samples to a reference spectrum, or is it better to run the standard each time with the sample?
Oh, okay. Good question. So um, every lab's quality, I should give a caveat to this. So every business, every company within the pharmaceutical space has their own quality standards, and so take what I say with a grain of salt. But I can say that I have seen a number of people who will collect one. They will have a standard material, and they will collect that piece of data. And then as long as you're using the same instrument and the same optics and the same experiment, the same scan parameters, it's very easy to sort of overlay a reference pattern versus, okay, so if we come back to, say, this example. So these two patterns could then serve as references for comparing batches of acetaminophen, right? So it's possible within EVA to simply then overlay patterns like this, and then you could stack them in, say, a waterfall plot or this offset plot. So you add a little bit of a Y offset for clarity. And then visually you compare them, or you would create a set of peaks, and you would say, yep, the strongest 7 or 8 peaks are present. They are in the right location. They are there in the right relative intensities. We can confirm that we have the right material. But is it necessary to run that standard every time? Probably not, but your own quality processes will generally dictate whether or not you need to do that for each batch. But
Fantastic. The next question for polymorph ID in a suspension, how comparable will the XRD of the sample look to a sample of the pure API?
a suspension of materials. So I guess I'm going to assume right now that, and please feel free to correct this in the chat if this is not the case, but I'm assuming that when you're talking about suspension, that it is predominantly API and then whatever the liquid state is. So a two-component slurry. If this is the case, what you would see is something similar to here. So you see how the background right there around 20 degrees is not perfectly flat. These samples were actually collected in a capillary, so you get signal from the glass. If you had, say, water or some sort of grease or oil or some sort of some liquid, you would expect to see a non-flat background. So some sort of gentle rise, maybe a single hump, maybe two. But then the comparison would be for the API in the wet suspension versus the API dry. So ideally, the peak positions and locations would be exactly the same. The difference would be predominantly in the background.
Fantastic. Another question is, how long does it take to collect good data?
That is a bit of a loaded question. Good data can be good data can mean a lot of things to a lot of different people, right? Just in the way that being tall is relative to who you're standing next to, right? So good data, I would look at the scans here shown and say, that's pretty good data. You want to have nice, sharp, skinny peaks, good signal in the background, good signal noise. You don't want to see a lot of visible wobble in the background. Here we can see, depending on how big of a monitor you're looking at, you can see the noise there. Something like this has even less noise, so this would be an exceptional quality scan. These are standard NIST standards, so of course I would expect them to give you very, very good data. But if you're aiming for things that are um, low noise, typically would be the determining factor for good for data would be low noise. 10 minutes to half an hour, depending on the amount of sample. There are a lot of caveats here, unfortunately, so it's hard to give an exact answer. Rule of thumb, I would say probably, I don't know, 10 to 15 minutes, maybe half an hour if you don't have a lot of material. But again, depending on which part of the pharmaceutical space you're working in, more sample will give you more signal. So if I have a lot of sample, I'm going to be able to reduce that noise a lot faster than if I only have a few little milligrams to work with. If you're doing that dusting method on the silicon wafer and you're working with very small amounts of material, taking a good data set might take half an hour to an hour.
Fantastic. Next question. If I am analyzing an unknown sample, how would I know whether the sample is crystalline or amorphous?
Okay, simple as this. Do you see peaks? Yeah. So for something like this, if I didn't know better and I weren't actually looking for it and I were looking at the red pattern, the only way that you see those peaks is if you're really, really looking for it, right? So these are pretty good quality scans on a predominantly amorphous material, but you see, I don't have a lot of peaks. Generally speaking, for crystalline materials, you'll know. You'll know because there will be some sort of signal that is not broad and wide. There will be some sort of sharp intensity. This is the data shown on the screen is, I guess, a little bit of a challenge case, right? We wanted to see how low can you actually detect it. By the time you're out here at, say, a couple of weight percent, so 3 to 4 to 5 weight percent, if you've got a crystalline phase, you know, because you see a sharp peak coming out of your background.
Fantastic. Next question is, sometimes we analyze formulation samples and we see significant interference from microcrystalline cellulose excipient.
Mhm.
How can we avoid interference in the analysis?
Well, so if it's there, it's there. Unfortunately, anything that you put in the sample holder or basically in between the X-ray tube and your detector is going to give you signal. So if you were looking at a formulation and there is a lot of microcrystalline cellulose in there, or any excipient for that matter, whether it be like lactose or mannitol or any of the other polysaccharides, you'll see it. And unfortunately, it's not so simple as you can't direct the X-ray to avoid the sample if it's in the sample holder. So a couple of different strategies that I've seen employed within the field, and one of them is physical separation. So something like this, right? So in this case, we had a physical shell, and what we did was we decided to remove it by shaving it off the outside. You could do maybe chemical separations. You could do maybe gravimetric separations. Those can give you a handle on trying to get just the other phases, but short of extracting out different phases, it's really hard to just to remove that effect, to remove all of this amorphous signal that you would get. So either you process the data even with this, and then you make a note that says all of this broad scatter is going to be from the cellulose. Maybe you try to do some sort of quantification off of it, and that becomes the useful piece of information: is how much excipient do we have in this particular formulation. But I would say if you are doing any sort of chemical separations, always run your sample first in its native state. Because if you do some sort of treatment, if you try to dissolve out your API and recrystallize it, you want to make sure that when it recrystallizes that you're getting the same form, so that you would see the peaks in the same locations as before. Because if you've done some sort of solvation and extraction and then recrystallization, if you're not careful, you might actually end up with a different structure than you started, or a different salt or a different precipitate.
Fantastic. I have another great question that just came in. For low percent crystalline detection, do you use signal to noise or another means to define the limit?
So this is also going to be dependent on your site. A lot of the diffraction quantification techniques have been derived from things like GC mass spec or traditional chromatography, where you would do a signal to noise calculation. So here, if you look on the left-hand side, I would figure out that I've got about 1 count per second from the baseline for that red peak, and then the wobble there in my noise. You set an upper and lower threshold for your noise. Unfortunately, in the case like this, because I have like the previous question mentioned, quite some significant contribution from excipients, and this is the true sample. There are a large amount of excipients here. Determining what your true background is a little bit tricky. But if I look at this region here, say around like 18.5 or 18.6, what I could do is define an upper and lower threshold for noise, take half of that value, and then do a calculation for signal to noise. Typically speaking, guidance is whether you're looking at limits of detection or limits of quantification. So LOD or LOQ would be either 10 to 1 or 3 to 1. So depending on what you're looking at. Yeah.
Fantastic. And I think we have time for one more question. If low crystalline signal is there, does increasing scan time help in increasing the signal?
I probably need to rephrase that question. So increasing the scan time will increase the statistical averaging of your sample. So way back earlier in this presentation, one of the things that I mentioned was that sample rotation is standard on all of the diffractometers that Bruker offers. And one of the reasons why you want to rotate your sample is because you are then effectively tumbling your crystals, right? If you think back to that video that we saw with all of the cartoons, the Bragg condition is fulfilled at very, very specific angles. So you really need to have a crystal oriented in the right direction. So even if you have, say here, like say, a quarter of a whey percent of crystalline API, that may only represent 1 or 2 crystals or little particles or grains in your whole sample holder, right? And if it's not positioned exactly right, that Bragg condition might not be fulfilled. You might fulfill a different Bragg condition, but it's going to be very, very tricky to satisfy that that 2 theta requirement, with the planes of atoms being positioned exactly perpendicular to the or parallel to the top surface of your sample. So somewhat rambling answer to come back to that is by spinning your sample, you can effectively increase your statistics by making it look like you have more average orientations of your crystal, right? So when you count longer, what you're doing while you're spinning is you're able to get more of these averaging times in there. Yeah. So when you look at, say, if we keep everything else the same, if we do a 5-minute scan, a 10-minute scan, and a 15-minute scan, but we don't change anything, we have the same sample, the same optics, the same amount of sample, everything is held the same except for the counting time. The 15-minute scan will have lower noise than the 10-minute scan, which will have lower noise than the 5-minute scan. But the signal to background is going to be exactly the same, because longer counting means you get more signal, but you also get more background. So it's kind of a the original question I think was like, do I get more signal by counting longer? And technically, the answer is no. You don't get more signal relative to your background because you count twice as long on your signal, but you count twice as long on your background. But what you do gain is a reduction in your noise. So your signal's noise will go up as you count longer. And I think that it improves as about, I think it's a square root function. So twice as long will give you not quite twice as good signal noise, 4 times as long, same thing.
Fantastic. All right. Well, that brings us to the end of the Q&A. Nathan, thank you so much for presenting today's webinar. To our guests, as a reminder, you're going to receive an email within the next few days with a link to this recorded webinar, so you can take a look at it later and review some areas of interest. We encourage you to visit lqa.com/resources for future webinar details and to access our library of recorded on-demand sessions. Thanks again, everybody, and have a great rest of your day.